Hello everyone and welcome to my channel. So today we are going to have a discussion on the topic of G protein coupled receptors or GPCRs. We will be covering our topic under the following headings. So let us take one heading at a time. A general discussion on the topic of GPCR. Now the GPCRs or the G protein coupled receptor, they are the commonest single class of target for the therapeutic drugs. In much more easy language, they are the commonest receptor on which a drug is going to act. The second thing to note is that once these receptors they have been activated by the agonist, a number of subcellular events will be occurring. So in a nutshell, if you want to summarize the event, the first step obviously will be when the extracellular ligand or the agonist, when it will be selectively detected by the cell surface receptor. After the receptor has recognized the agonist, then there will be the activation of the GTP binding protein, also known as G protein, which is located on the cytoplasmic phase of the plasma membrane. Now, one of the most important mechanisms by which the G protein coupled receptors they act, that is by the generation of the second messengers. And these second messengers, they actually form a pathway which is known as the signal transduction pathway. What happens in signal transduction pathway is that a signal, suppose it is reaching the receptor, now that is going to activate another compound known as the second messenger. This second messenger is going to activate another compound. This is going to activate suppose another compound, and ultimately there will be the effect. So the signal is passed down from the second messenger to the subsequent messengers for the effect to occur, and in between this the signal gets amplified. So what are the G protein coupled receptors or GPCRs? Now these GPCRs, they are a large family of receptors that have a 7 transmembrane alpha helical motif structure. They are also known as serpentine receptors. Why serpentine? Because the receptor polypeptide chain, it just looks like a snake when it crosses the plasma membrane. And how many times it crosses? It crosses the plasma membrane 7 times. This receptor has got 3 extracellular and 3 intracellular loop. And the agonist binding site, it is located somewhere between the helices on the extracellular surface. Now let us have a look at the structure of the G protein coupled receptor. Now this G protein coupled receptor, as we have just now studied, that it is a 7 transmembrane receptor, right? So 1, 2, 3, 4, 5, 6 and 7. Why transmembrane? Because it is crossing the entire plasma membrane. A portion of the receptor is located outside the cell. Another portion is located inside the cell. The outside of the cell, it has the N terminus, and the inside of the cell, it has the C terminus. But we also saw that this receptor has got 3 extracellular loops. So these are the 3 extracellular loops and 3 intracellular loops, 1, 2 and 3 intracellular loops. In between the loops on the extracellular surface is the point where the agonist is going to bind. Once agonist is bound, a number of events is going to occur at the subcellular level. What are the events? Now we will see. Now one single GPCR, it can utilize more than one G protein, and this concept is known as agonist pleiotropy. In the humans, we have got 800 GPCRs. Among those 800 GPCRs, currently we have got 130 GPCRs that act as a target for the therapeutic drugs, either as an agonist or as an antagonist. You have got 130 which are orphan receptors. What do we mean by orphan receptors? Orphan receptors means that basically we don't know what the ligand of the receptor is. And also we don't know what the function, exact function of that receptor is. And 420 they are sensory receptors, most of which are olfactory. Before moving forward, let us see what is the structure and the action of the G protein coupled receptor. Now this G protein coupled receptor, it basically couples to the heterotrimeric GTP binding regulatory proteins or the G proteins. So since it is regulated by a GTP binding protein, that is why it is known as a G protein. And since it couples with that, it is why known as coupling receptor. So the entire name of G protein coupling receptor comes from the name of the regulatory protein. Second thing to note is that it is heterotrimeric in nature. Why heterotrimeric? Because it has got 3 different subunits: alpha, beta and gamma subunit. In the inactive state, what is happening? Let us suppose that this is a G protein coupled receptor. Let us suppose this is the extracellular component, this is the intracellular component. So when this receptor is in the inactive state, then you have got 3 subunits, as I have already told you, the alpha, the beta and the gamma. To the alpha subunit, GDP is bound. So this is what is happening in the inactive state. Now once this receptor is getting activated, so let us suppose that this receptor, the G protein coupled receptor, is getting activated by an agonist. So in the inactive state, GDP was bound to it. Now once it is activated by the agonist, G's GDP, it will be removed. And in place of GDP, we are going to get GTP. Right? So once it is activated, this G protein coupled receptor, it promotes the exchange of GDP for GTP. And where is it occurring? It is occurring on the alpha subunit. And once this alpha subunit, you had 3 subunits, one was alpha, one was beta and one was gamma. So now after it has been activated, the GDP has been removed and GTP is present now in the alpha subunit. So once this happens, once GTP comes to the alpha subunit or binds to the alpha subunit, the GTP and the alpha subunit complex, it basically dissociates from the beta gamma component. So now you have got two components. One component is where you have the alpha and the GTP. The second component is where you have the beta and the gamma subunits. Now this alpha and the GTP subunit, this is the component that is going to mainly activate the second messenger system and help in the signal transduction. Also the role of beta and gamma receptor is also there, that we are going to study a little bit later. So as I was saying that this is the inactive state. In the inactive state, what is happening? You have got the G, alpha, beta and the gamma, all the three components of the heterotrimeric protein, it is present. And on the G alpha component, you have got GDP bound. Once it is activated, the G protein coupled receptor is activated by the agonist, immediately the GDP will be replaced by GTP, and the G alpha and the GTP subunit, it is going to dissociate from the beta gamma subunit, and this is now going to activate the second messenger system. The question that arises is that why will GDP be replaced by GTP? Now, let us suppose this is a G protein coupled receptor. Now, I am not drawing the entire structure. Let us suppose this is a G protein coupled receptor. Now, we know it has got a extracellular extracytoplasmic end and an intracellular end or intracytoplasmic end. In the extracytoplasmic phase, the agonist is getting bound. So once this agonist is bound, what is happening is that you have got 7 transmembrane helical structures. So let us suppose this is helical structure number 6. This is the 7 transmembrane helical structure. Okay. So once this agonist is bound, these helical structures, they are going to separate from each other by a distance of 1 nanometer. So if they separate by a distance of 1 nanometer, it is going to open up a cavity in the receptor cytoplasmic surface, right? Suppose this is also moving 1 nanometer, this is also moving 1 nanometer. So now this cavity, it is going to increase. So once this cavity increases, suppose this is where the GTP, where the GDP was bound. So now the binding will be loose. So naturally, it reduces the nucleotide affinity for the G protein. So since it is loose, now the GDP is being bound loosely. So now that is going to dissociate and the GTP is going to come in its place. Why? Because in the cell, the concentration of GTP is more than the concentration of GDP. So if the GDP is getting dissociated, obviously the GTP is going to come and take its place. The next question that arises is what is happening to the beta and the gamma subunit. Now the beta and the gamma dimer, it basically activates the receptor operated potassium channels. It also so what is going to happen if the receptor activated potassium channels are getting activated? There will be hyperpolarization. It is going to inhibit the voltage gated calcium channels, and also this beta and gamma subunit, it promotes the G protein coupled receptor desensitization. Once there is high rate of G protein coupled receptor activation. Let us see what are the types of G protein coupled receptors. So you have got mainly 5 types of G protein coupled receptor. The GS, the GI, the GO, GQ and the GOLF or the G olfactory. Now these are those G proteins that are bound to the alpha subunit. So based on the type of G protein that is bound to the alpha subunits, the effects are going to vary. Suppose if it is of a GS kind of a protein, as is found in the beta adrenergic amines, in the histamines, in serotonin and for glucagon, for them what is going to happen is that adenylyl cyclase will be activated. So once adenylyl cyclase is getting activated, there will be increase in the level of cyclic AMP. This increase in the cyclic AMP, it is going to increase the calcium channel opening. If it is of a GI kind, as is found in the alpha 2 adrenergic amines or for acetylcholine, the muscarinic receptor, for opioids and for serotonin, it is going to decrease the adenylyl cyclase activity. So because of the decrease in the adenylyl cyclase activity, there will be decrease in the cyclic AMP and hence the calcium channel opening will be inhibited. Also, it opens up the cardiac potassium channels. So there will be hyperpolarization and ultimately there will be decrease in the heart rate. That means GS, it is stimulatory, whereas GI, it is inhibitory. So this GO, it is mostly found in the neurotransmitter of the brain. The exact areas are not known. It causes the inhibition of the calcium channel. So what GQ is doing? GQ is also found in the acetylcholine muscarinic receptor for the bombesin and for the serotonin, the 5H2 kind of receptor. It basically increases the activity of phospholipase C. And because of increase in the phospholipase C, action of IP3 and diacylglycerol, they are increased. Now we know that this IP3 and diacylglycerol, this acts as the second messenger. And this actually helps in removing the calcium that is stored in the sarcoplasmic reticulum and brings it to the cytoplasm. So there is increase in the cytoplasmic calcium level. The GOLF or the G olfactory, it is mostly found in the olfactory epithelium. They are odorant kind of a receptor. And this is also stimulatory kind of receptor because it increases the level of adenylyl cyclase and finally the level of cyclic AMP. Now what are the pathway for the G protein coupled receptor action? So the major pathways through which the GPCRs they function, it is the first is the activation of the adenylyl cyclase. So once adenylyl cyclase is being activated, there will be increase in the level of cyclic AMP. And this cyclic AMP we know, it functions as the second messenger. And finally it is going to increase or rather open the calcium channels. Also cyclic GMP can act as a second messenger. Another pathway is activation of the phospholipase C. Once phospholipase C is getting activated, IP3-DAG pathway will be activated. This IP3-DAG pathway will act as the second messenger. And finally it is going to release the calcium from the sarcoplasmic reticulum. And this calcium will be released into the cytoplasm. And there will be increased intracytoplasmic calcium. Also, some of the ion channels are G protein gated. And two most important channels are the inwardly rectifying potassium channels found in the heart. And also the voltage gated calcium channels. Now, how is the action of a G protein coupled receptor getting terminated? The alpha subunit, remember, that was dissociated. This alpha subunit, it has a GTPase activity because upon activation GTP was bound to the alpha subunit. So because of this GTPase activity, the bound GTP, it will be slowly hydrolyzed to GDP. So once this hydrolyzation takes place, the alpha subunit, it then dissociates from the effector molecule and joins with other subunits. What were the other subunits? The beta and the gamma. So the alpha subunit is now getting bound in place of GTP to GDP and rejoining the beta and the gamma. That means it is coming to its resting state. But in between that, what has happened is that the effector molecule, or suppose the second messenger, it has been activated or either inhibited. And this activation or inhibition is going to occur for several seconds, which is much longer than the lifetime of the activated receptor, which is in milliseconds. So because of this effector molecule activation or inhibition for larger second, the signal has been greatly amplified, or as we call, there was the signal transduction. Now there is another component which is known as the RGS. This RGS is also a protein. So this RGS is a regulator of the G protein signaling. And what is its action? This RGS actually regulates the rate of the GTP hydrolysis by the alpha subunit. And thus it also determines the period for which the G protein coupled receptor is going to remain activated. So to sum everything up, GPCRs, they are the commonest single class of targets for therapeutic drugs or the commonest receptors. The activation of the receptor, it will set into motion a number of subcellular events. The first step will be the extracellular ligand is selectively detected by a cell surface receptor, and once the receptor is getting activated, then it triggers the binding of a GTP binding protein, the G protein, and this happens on the cytoplasmic phase. Another important mechanism for GPCR is the generation of second messengers, and the types that we have studied are GS, GI, GO, GQ and G olfactory. So that's it for today, guys. If you like the video, then give it a thumbs up and share it. Also subscribe to my channel and please press the bell icon so that you don't miss out on any upcoming videos. Also let me know down in the comments below what other videos you would like to watch. Until next time.